Good evening, everybody. I just wanted to take a moment to welcome you all to tonight's symposium. It's going to be an exciting evening. My name is Mike Clagg. I'm the Dean of the school. And as everybody knows, this is a symposium in honor of George Graham. And in honor of him, but then in honor also of two outstanding individuals, our own Carl Taylor, and I guess Nevin Scrimshaw is in a taxi, right? And hopefully it's a taxi who knows where our school is. So my only job then after welcoming you is to thank everybody who was involved in this evening and making it a success. Our folks from External Affairs, Monique Forbes-Dickerson and everyone else. And to acknowledge and compliment Keith West for putting this together. Keith, as everybody knows, is a professor in our school, but not just any professor. He's the George G. Graham Professor and a former student of Dr. Graham's. So he's our first speaker for tonight, and I'd like to welcome Dr. West to the podium. So.
Thank you, Dean Clagg. Allow me to extend a very warm welcome to everyone here, distinguished visitors, fellow faculty, students, staff, guest speakers. It's a very special evening for a lot of reasons. It's an intersection of many points in time. It's the third Graham Lecture in honor of George, but it's also the inaugural endowed lectureship in Dr. Graham's name that is been made possible by a generous grant from the Mittendorf Foundation here in Baltimore. So that's special all by itself. And then it happens to be the 40th anniversary of the publication of WHO monograph on nutrition and infection, interactions of nutrition and infection, that was published by Nevin Scrimshaw and Carl Taylor and John Gordon 40 years ago, plus or minus some standard error here. It was in 1968. As Klaus Kramer, I was speaking with him from Sight and Life in EB a few days ago, reminded me, because I was lamenting that we're really 1 year off, and he said, don't worry about it, it's the 50th anniversary of the publication, the 1959 publication in the American Annals of Medicine on the same title of nutrition, interactions of nutrition and infection. So it's a 40th plus or minus, and it's exactly, it was March of 1959, it's exactly the 50th anniversary plus 23 days of the anniversary of the publication of that lead manuscript. So I've probably gone off of my slides already because, you know, I get up here and I start to chat, and I'm so excited I can hardly speak, as it were. I've been told that I sound like Barry White today, that I should be speaking at some club rather than here. But let me say a few words about Professor Graham. He was the professor of pediatrics and international health at the School of Medicine and here in the School of Public Health. He was the founding director of what was for years the only division of human nutrition. It was the only division in the Department of International Health for a number of years. He was the first professor of human nutrition within the department. He founded the Institute of Nutrition in Lima, Peru in 1961, where he was working for years beforehand at the British American Hospital, and realized that there was a lot of malnutrition that needed to be addressed, that needed to be understood, that needed to be treated, and needed to be prevented. And so he set about to set up the Institute of Nutrition in Lima, and as its founding director, which today stands as one of the leading nutrition institutes in the world, and from where one of our speakers this evening, Dr. Claudio Lanata, is a professor and has traveled all the way from Lima to be with us this evening. Dr. Graham received many awards and had appointments on advisory boards. He was known for his opinions. He was outspoken, he was passionate, and his insistence on evidence doing the talking rather than people earned him many accolades and many friends and many enemies and many people in between. You had to listen to Professor Graham when he spoke because he had authority behind his voice. He was a member of the National Academy of Sciences. He was on boards with the NIH. He was on many different boards of the American Institute of Nutrition. He gave congressional testimonies about the dangers of America becoming obese long before the current epidemic that we're facing. He was a voice calling in the wilderness at that time, saying that America needed to change its eating habits in the 70s, so it would not be facing an overweight epidemic in the new millennium. He received the Joseph Goldberger Award from the American Medical Association, the Borden Award from the American Academy of Pediatrics. These are well-known nutrition awards, as well as the E. Leon Garcia Award from the Pediatric Society in Peru. He was a teacher, a mentor. He was very concerned about the school, about the Department of International Health, the Division of Human Nutrition. He had a great deal of foresight, and he was the world's leader in figuring out how to feed infants and young children. His research in Peru stands even today as citable references 30 years later, 40 years later, about the consequences of early malnutrition and the benefits and the interactions that are observed when young children are fed different kinds of diets in their infancy and early childhood. He published over 250 publications. So he is one of our Hopkins, he's one of our tall trees in the Hopkins forest. And we have a legacy that we are building in his name, beginning with the installment of the George Grant Professorship in 2005, for which he was here, from which funds from family and friends of him allowed that to happen. And it is a lasting tribute to his contributions. We've had the George Grant Professorship Lecture. I was told I could not call it a lectureship when we started in 2006 because it was not endowed. It was coming out of the professorship funds. And so it had to be called a professorship lecture, and that's what it was for the first two lectures given by Ken Brown in 2005 and Bill McLean in 2007. But now we have a slightly different landscape. We now have a lectureship in his name, and this is another tribute to his stature and contributions here at the school. So this is the first year for that occasion, and it's a wonderful event to be then commemorating the 40th anniversary of the WHO monograph on interactions of nutrition and infection. That has been published in a number of languages over the years and really has served as a platform for understanding what we knew about this interaction up to that point in time, and it has served as a source of vision for guiding the research over the past 40 years and beyond. It was one of those special publications that we have all read and been familiar with, and we're very thankful for it being written. And I just point out that it was really 50 years ago that the first publication with this title came out from Scrimshaw, Taylor, and Gordon. So we have Charles Stevenson talking about the effects of malnutrition on infection, the first part of this vicious cycle, as it were, who is a research scientist at the USDA Western Human Nutrition Research Center and an adjunct professor at the University of California, Davis. We have Claudio Lanata talking about the effects of infection on nutritional status of the host. Claudio is senior researcher at the Institute of Nutrition in Lima, Peru. I decided to keep it in English because I would destroy the Spanish. So he's going to complete the other part of that circle. And then we have our own Professor Bob Black, chairman of our Department of International Health, and Edgar Berman Professor, also the director of the Institute of International Programs, who's going to bring those two paths together and try to give us a little bit more vision of the way forward and how we can make this area of interactive interactions between nutrition and infection continue to be a vibrant area for research in the future. Then we will have a conversation with Nevin and Carl. Nevin, I won't go through all this other than to say that Nevin is truly one of the, if not the father of modern nutrition today. He has published over 650 papers. He's been the founding member of a number of institutions with the UN University. He is the editor-in-chief of the Food and Nutrition Bulletin. The list goes on and on. He's one of the greats in modern science and nutrition. He started the Institute of Nutrition in Central America and Panama, INCAP, which has for years published numerous papers about the importance of nutrition and child health. And we're just delighted that he's coming. He's not here yet, but he's coming. He's a laureate of the World Food Prize and so forth. So when he comes, I'll be much relieved when I see his face, actually walk through the door. And then we have Carl Taylor, who is the founder and first director of our Department of International Health. And if I got that right, Tim? I think, yes. We've got Carl, who is the first director and founder of our department. It's the first department of international health of its kind in any school of public health in the world. And so it all started here. Life begins here in public health and in international health. And the students every year feel that, I think. Dr. Taylor worked for 30 years in India as the director of the Presbyterian Mission Hospital. He was the founder and the lead researcher for the Narangwal Project, which set the stage for how we understand nutrition and infection interactions in the South Asian context. We've learned enormous amounts. We studied the Narangwal Project when I was a mere student here in the late 70s and early 80s. Carl was the director of the chair of the department from up until 1983, 84, at which point Bob Black took over. And Carl went to China to direct UNICEF, where he spent the next 4 years leading the UNICEF charge in China. He's the recipient of numerous awards, including the U.S. Presidential Award for service to children in the world who are in especially difficult circumstances. He's co-authored a book with his son, Daniel, who's also here. We're delighted that Dan is here with Carl, called Just and Lasting Change. That was published by Johns Hopkins University Press. He's a teacher and a mentor to thousands of us who are in international health, and we are thrilled that Carl is here as well. I'd like to also just finish by saying that tonight's proceedings are going to be incorporated into a special commemorative book on interactions of nutrition and infection 40 years later or 50 years later, whichever the title, whichever way we go, along with the first chapter of the monograph that was published 40 years ago. I'd like to welcome the Grant family here. Carol, welcome. It's very nice to have you. These proceedings are being videoed, so we'll have this up on YouTube probably by 10 o'clock tonight. We'll have this book that captures the early forward, a later forward that Carl and Nevin are going to write, the papers from Charles and Claudio, plus the comments of the evening, so make some good comments and questions. And they'll be published by Sight and Life. We have permission from WHO to republish portions of the original WHO monograph in as a special commemoration of this evening and the event itself. And I'd like to recognize Dr. Klaus Kramer from Sight and Life. Sight and Life is publishing this all for us, and if you fill out your card and drop it in the box at the end of the evening, either here or upstairs, you will be sent, when it comes out, a free copy of this commemorative publication. Sight and Life is also kindly agreed to put the proceedings into a special proceedings issue of the Sight and Life newsletter, which reaches about 10,000 people around the world. So we'll start off the evening with Charles talking about malnutrition and infection, and then we'll round it up with Claudio talking about the role of infection on malnutrition, and then catch this, Bob. We're going to have Bob bring it together. I actually tested that beforehand. I was afraid the hours were going to go the other way. And then we'll have a conversation with Nevin and Carl. These are supposed to be lazy boy chairs, where we were going to have a floor lamp and soft lighting with paintings coming down to make it look like a living room. But this is as close as we've got to a living room. And we'll enjoy a conversation with Carl and Nevin, and then a reception at the top of the hop on the 9th floor. And there are surprises up there as well. So everyone is invited to the 9th floor for a very, very nice reception, Peabody Wind Ensemble, wine, and so forth. I have stopped my comments at this point and would like to welcome Dr. Charles Stevenson. Charles, where are you? Here you are. To the podium to talk about the first half of this vicious cycle.
Oops, static electricity. Well, thank you, Keith, for that introduction, and I'd like to thank Johns Hopkins and the Bettendorf Foundation and the George Graham Lectureship for inviting me to sort of be the lead-off batter here tonight. And thank you all for coming. I remember, so I was a student here about the same time Keith was, and the last time I was in this building, or in this room, it looked quite a bit different, and I think I was listening to Leon Gordis talk about hepatitis A, is my last memory of this hall. So this is my topic, and since I'm going to talk about effective malnutrition on resistance to infection, we do need to start with a little primer on the immune response to infection. And this graph is out of one of my favorite books published in the mid-70s, Pathogenesis of Infectious Disease by Cedric Mims. And it shows on the top there the infection. Oops, let me try the pointer. So this represents infection by a mucosal pathogen that may be short-lived and cleaned up by the innate immune system, represented at the bottom by that monocyte, which will differentiate into a macrophage, and this neutrophil. And then here we have bacterial or viral replication. And one of the things I noted about in reading the monograph is that there were a lot of very well-conducted animal studies, and I think they had three common characteristics. One, they were good, solid nutrition interventions, with some of them with single nutrient deficiencies, for example, pair-fed controls, lots of different pathogens, lots of different animal species, good microbiological endpoints and pathological endpoints. So at the time of the publication in 1978, much of the information here, so an initial infection, perhaps through a process of invasion through mucosal surfaces, becoming a systemic infection, and also showing virus replication sort of systemically. This sort of information was well developed, and we knew a lot about what specific deficiencies could do in animal models. And I think there were 53, vitamin A is my favorite vitamin, and I think there were 50-some vitamin A studies. Nowadays, what we've, since 1968, we are focusing on this part of the graph, I guess. Can you, yes, it is up there behind me, I can see it. the immune response to infection. And we knew relatively little about cellular immunology at that point, and we know a lot now. And so the experimental animal studies tend to reflect that. And so this evening I'm going to talk about innate immunity and adaptive immunity. And they're not really two separate systems, but adaptive immunity we defined in the 60s, 70s, 80s, and identified, for example, T lymphocytes and antigen-presenting cells, which present antigen to T lymphocytes, which provide help to B lymphocytes and produce antibody. And the innate immune system we've learned a lot more about since the sort of early 90s. And we knew about these phagocytic cells, which were discussed, well, have been discussed since the early 20th century. And data on the effect of malnutrition on these cells was presented in the 1968 monograph. But now we know a lot about how these cell types, including other actors in innate immunity, which can include epithelial cells at mucosal surfaces, other stromal cells, cytokines, chemokines, lipid mediators like prostaglandins and leukotrienes. There's a whole network of mediators of inflammation that are triggered by the initial response and recognition of a pathogen by a specific group of pattern recognition receptors in the innate immune system that we didn't know anything about until the 1990s. So this immune response to infection, which here is coming up around day 5, really starts about an hour after infection and recognition of the pathogen by the innate immune system and triggering inflammation. And then the innate immune system really links to the adaptive immune system and triggers the memory response. So I'm going to differentiate a little bit and show some examples between the innate and adaptive immune system and effective nutrition on these different cell types. So in deference to Keith's preoccupation with 40 years, I stopped my review of the literature in 2008, as indicated on this slide. He was worried about that months ago. So Chapter 3 and Chapter 4 in the monograph really describe the effect of malnutrition on resistance to infection in Chapter 3, which is big and it has several very long tables. And Chapter 4 is really talking about immune function, and it's very slim. So there's a description of synergism and antagonism between nutrition, malnutrition, and a pathogen. So synergism would be increased severity of infection with malnutrition. But there's little mechanistic data. And so as I've already mentioned, a current focus, particularly in animal studies, not so much in human studies, is on mechanism. And mechanism of immune function, but we also know a lot about mechanism of action of nutrients that we didn't know anything about 40 years ago. And much of the data came from observational, clinical, and community studies, whereas I think a major advance between then and now is the application of randomized controlled trial methodologies in the community and the clinic to look particularly at specific, well, I was going to say specific micronutrient deficiencies, but at least specific nutrition supplementation, so vitamin A, zinc, multiple micronutrients, and so on. So that's a big difference now and has given us a lot of valuable information. And I mentioned the earlier animal studies, their endpoints, and now we're more focused on mice than a diversity of species. We can knock in or knock out genes of interest related to nutrient metabolism or immune function. And so, again, a lot of mechanistic studies are going on. So there's not quite a synthesis of that information yet. And very briefly, Chapter 4 talked about immune function, as I mentioned. And so on the right there in 2008, you can see we have B cells, cytotoxic T cells, various subsets of T helper cells that are pro-inflammatory in response to specific types of pathogens, or T regulatory cells that downregulate inflammation, gamma delta T cells, and the list goes on. And then, as I mentioned, the innate immune system, we know a lot more about that just in the past 15 years than we did 20 years ago. And one other point I wanted to highlight is, so on the left here you see protein energy malnutrition impairs protective immunity. That's clearly shown in the 1968 monograph. But we've had a nutrition transition that Keith alluded to earlier, so that now we're not only interested in sort of undernutrition, malnutrition, but we're also starting to think about, instead of protein energy malnutrition, the idea of dietary restriction sort of in a calorie-rich environment. So that's a difference in focus, and that leads to a difference, another difference, and maybe this is cheating a little bit, but we've been focusing on infectious diseases, but now we're also interested in chronic inflammatory diseases, which also obviously involve the immune system. So I'm not going to be comprehensive, and so this is an idiosyncratic view of sort of what I think the differences are then and now. And so I really picked 3 examples. And one example that's a change in thinking for all of us, I think, is that some nutrients that previously were thought of as calorie sources are now being differentiated and analyzed, and fat, generically fat, does more than provide energy. Fatty acids can modulate inflammation, and so I'll show you an example from the innate immune system. So this is a circa 1970-ish view of the macrophage in black, which phagocytoses a foreign body or a pathogen, kills it with lysozyme and reactive oxygen, and that can be impaired by malnutrition. But part of the revolution in innate immunity is identifying the mechanisms by which bacterial lipopolysaccharide can activate macrophages. We knew monocytes were activated to become macrophages. We didn't know how. They activate pattern recognition receptors. Toll-like receptor 4 is the one that's activated by LPS. And it regulates transcription of genes like TNF-alpha, IL-1 beta, and cyclooxygenase 2, which itself then metabolizes arachidonic acid to leukotrienes, well, to prostaglandins. And these are mediators of inflammation. So just as an example of how fat is no longer just calories, this is an in vitro study that shows that saturated fatty acids themselves can actually activate toll-like receptor 4. And this, if you think about lipopolysaccharide, it has a lipid component of short-chain fatty acids, lipid A, so it itself has a lipid component. But this experiment shown here from Dr. Dan Wang's lab shows that the toll-like receptor 4 in the red box at the top on the left is activated by LPS, but it can also be activated by saturated fatty acids, or saturated fatty acids can enhance the activation of LPS. And there's a lot of complicated immunology in the cytoplasm there, but if you get down to the, so I'm going to skip that. But if you get down to the nucleus, the activation of toll-like receptor 4 is activating gene transcription, in this case COX-2. So COX-2, if you put a reporter construct in a cell line that is expressing toll-like receptor 4 as a model system, activate it with LPS, a low level of LPS, you get 100% of, this is driving expression of luciferase. So you get 100% expression. This is enhanced by saturated fatty acids and can be diminished by polyunsaturated fatty acids. So in addition, so in this case, the dietary fat may be acting as a pathogen. So this is an excursion into the area of chronic inflammatory disease. Another activity of polyunsaturated fatty acids of marine origin, so long-chain omega-3 fatty acids like EPA, shown in the blue on the right, is it can be a direct competitor of another essential fatty acid, arachidonic acid, which is the precursor through the COX and LOX enzymes of 2 and 4 series prostaglandins and leukotrienes that mediate inflammation. So the membrane levels of these 2 fatty acids depend entirely on diet. So if your intake of EPA goes up, the composition of the membrane in this granulocyte changes, and when these enzymes are activated, there's a precursor now, not only arachidonic acid, but EPA. And EPA-derived 3 and 5 series prostaglandins and leukotrienes are much less inflammatory. And also, because of the competition, there's fewer 2 series prostaglandins and 4 series leukotrienes produced from arachidonic acid. So EPA can be anti-inflammatory in this context. And there's a lot of clinical trial data showing that in chronic inflammatory disease, for example, arthritis, if you have granulocytes in your joints, you can take EPA from fish oil supplements. Pet foods now have EPA in fish meal components, fish oil components for the same reason. You can decrease leukotriene production, which can subsequently decrease production of pro-inflammatory cytokines. So if you look at the percent of EPA in the monocyte membrane, it's inversely correlated with production of IL-1 beta and TNF-alpha. So this is a good thing for inflamed joints. If you go too high, this will decrease the bactericidal activity of these same cells. So there's a plus and a minus. So protein energy malnutrition, example 2. I hope someone's timing me. Keith, okay. So I have another hour or so. I'm doing okay. Okay. Protein energy malnutrition and dietary restriction, as I mentioned earlier, can affect adaptive immunity. So I'm going to talk about T cells in the thymus, where T cells come from. So we know, and knew in 1968, that protein energy malnutrition impairs T cell mediated immunity. I guess we didn't know it was T cell mediated necessarily then. But in children with malnutrition, if you can image the thymus in infants, it's smaller. The number of recent thymic emigrants, new naive T cells, is lower. The T cell areas of lymph nodes is smaller. And if you look at a T cell mediated response like delayed type hypersensitivity response on the right, you can see a lower response in kids with malnutrition. That's data from a work in Ghana, I believe, of Charlotte Newman in the early 1970s. So the kids with the worst malnutrition have the lowest DTH score, area of induration there. And thymic function and this DTH response can recover pretty quickly when malnutrition is treated. So what's the mechanism here? And these are data from a paper from 1998 in Nature that shows that T cell immune deficiencies is induced by starvation. Okay, we knew that. But also reversed by the hormone leptin. So leptin is a hormone that's involved in sort of long-term energy balance. It's produced by adipose tissue. If you have lower adipose stores, you have lower serum leptin levels, and so it reflects energy reserves of the body and is lower in protein energy malnutrition. Mice without leptin or its receptor have thymic atrophy. T cells have leptin receptors, and leptin promotes T cell survival, proliferation, and differentiation. So on the top left, figure A there, those are human lymphocytes in culture, and the bottom two lines are no or a very little bit of additional leptin, 100-fold more for the middle line, and the highest line is the highest level of leptin. So you alleviate the leptin deficiency in cell culture and you get better lymphocyte proliferation. On the right-hand side, you see an experiment in mice. On day 0, there are 4 groups of mice, and the mice were starved for 2 days, and that's body weight. So body weight drops by almost 30%, and then recovers rapidly by day 4 or 5. And then during that weight loss period, animals were immunized to induce a delayed type hypersensitivity skin test response, which we saw is diminished in children with PEM, and later challenged on day 6 or 7. And then the other intervention was leptin or sham treatment during the sensitization period. So the bottom bar graph shows on the far left, this is just our negative control, well-nourished but no immunization, so no DTH response. This is our positive control, well-nourished, robust DTH response. This is the starved animal without leptin, so diminished DTH response, then leptin treatment restores that response. So T cells respond to energy reserves. T cells and other cells in the body also have receptors for example, amino acids, and at least in studies with avian species, chickens, the thymus is reduced and T cells are diminished in number by protein malnutrition. but bursal cells, B cells are not, and it seems that the T cells don't upregulate a high affinity receptor for amino acids, and so seem sort of predestined under protein restriction to not fare well compared to other immune cell types. In humans, these are data, historical data, really looking at survival rates of children in the Gambia from Sophie Moore and Andrew Prentice, and kids that were born in the harvest season, birth weights are good, or in the hungry season, birth weights are low. Same mortality or survival curves here, but when they hit 20 years of age, the hungry season, now adults, have a higher mortality rate. If you look at a study of infants from 1 to 50 weeks of age, imaging the thymus, you see that kids, this is hungry season minus harvest season. So if you see a negative, that's a smaller thymus in the hungry season. So thymic deficits early in life. On the bottom, fewer numbers of recent emigrant T cells circulating in the blood. So definitely a thymic impairment. And so immunologists would suspect this later difference in mortality might be due to impaired adaptive immunity, but we don't have those data. But if we jump to sort of our current concern with obesity and look at some non-human primates that are sedentary and overweight, and we put them on a calorie restriction diet, the black bars, and look at the same endpoints. So these are males and females. So if you look at one or the other, the percent of naive T cells, so recent emigrants from the thymus, is now better in the calorie restricted group than the sedentary obese group. If you look with another method at recent thymic immigrants, you see the same thing. And if you see T cell receptor heterogeneity, it's better in the calorie restricted group, which means you have a better repertoire of T cell receptors to respond to different antigen types. So that's a good thing. So this is sort of, Aristotle would be happy, I guess, the golden mean. Too much is bad, too little is bad. And so we're thinking more about balance now than deficit and adequacy. And finally, Keith, I'm getting to vitamin A, and I'll wrap up in 3 minutes. So vitamin A, and again, this is an example, vitamin A serving as an example. So I want to mention that there are community intervention trials that have given us a lot of data. We have new mechanistic information. We know how vitamin A works in the immune system. And we're looking at adaptive immune endpoints. And vitamin A, the active metabolite retinoic acid, affects antigen-presenting cells, T cells, and B cells. And in 1986, Dr. Summer and West and other co-authors from this institution published a study that showed about 30% lower mortality in Indonesia with vitamin A supplementation compared to control villages. They were not placebo-controlled, and that observation was seminal and led to several placebo-controlled intervention trials, the results of which are shown here. So the lower relative risk of death in the vitamin A supplement group compared to placebo group from primarily infectious diseases in infants and young children. Same thing has been seen in clinical trials with some infections but not others. So this shows that the mortality from measles on the top right graph there is lower in the vitamin A group, the red bar. The duration of post-measles pneumonia is lower in the bottom graph. However, this is not a universal observation in that anybody with pneumonia is necessarily going to improve. And I mention this just to highlight the fact one thing I think we need to do is look at mechanism, particularly in human studies more. And this is a study that done in Peru that basically showed, so I was involved, others in this room were involved. Vitamin A supplementation slows recovery in children hospitalized with pneumonia. They all got out of the hospital at the same time. But if you look at blood oxygen saturation in the let me get these right, the vitamin A group, it recovered more slowly after hospital admission than the placebo group. The percent of children with retractions reflected that, and the need for supplemental oxygen. So a final word about mechanism, and this is just to highlight a little bit of data from animal studies. And 1987, Pierre Chambon and Ron Evans, working separately, identified the retinoic acid receptor. This is the receptor. We knew about retinoic acid, so as of 1987, we knew how it worked in the immune system and in other cell types. So in ex vivo cell culture studies with primary cells, you can take naive T cells, shown here, let them interact with antigen, and they'll develop a phenotype. A T helper 1 cell protects against intracellular pathogens. This is a big oversimplification, forgive me. Th2 cells will defend against extracellular pathogens like Vibrio cholera, for example, or respiratory viruses, and promote the IgA response. And just in brief, retinoic acid, indicated here as RA, can act on an antigen-presenting cell to decrease IL-12 production, which drives Th1 development. So that's vitamin A is diminishing a Th1 response potentially. Retinoic acid itself, if you look at the Th1 cell, can decrease interferon gamma production. Interferon gamma can downregulate down here Th2 development. And there's some other direct effects of retinoic acid on this T cell that drive Th2 development. So one of the responses driven by Th2 cell is a secretory IgA response at mucosal surfaces to a pathogen like Vibrio cholera, or in this case, influenza A virus infection in mice. So vitamin A deficiency, as you now expect, would decrease and high vitamin A increases influenza-specific salivary IgA response, and shown here. So this is the flu-specific salivary IgA, and it's really because there are fewer IgA, vaccine-specific IgA cells in the submucosal, in the salivary gland in this case. So it's really impaired the cell-mediated immune response. And I guess just one final cautionary comment. Again, I guess I'm always looking for the black cloud surrounded by the silver lining. But if you think about balance in the immune system, if you're improving some response, you're potentially causing pathology in another situation. So in a mouse, simple mouse model of asthma that's driven by Th2 cells, vitamin A deficiency will actually decrease, and high vitamin A increase the severity and increase the number of eosinophils, increase IgE, increase IL-4 and IL-5, and so on. That's not by any means a perfect model of asthma. So in conclusion, I guess the one thing I think about is if you're thinking about deficiency, so nutritionists have all seen this slide. This is level of vitamin A intake. Deficiency we're commonly defining by risk of impaired retinal function. We now know there's increased risk of infection or death from infectious diseases. At least in some subgroups, it may be worth looking at immune dysregulation as a potential sort of toxic or high-level side effect of vitamin A and some other nutrients. So, as I've said, the nutritional deficiencies, as we learned from the monograph in 1968, are definitively associated with severity of infectious diseases, risk of death. And since then, we've learned a lot of things. So just the three I'd highlight is that the randomized controlled intervention trials have shown us a lot about single nutrients. We have a lot of mechanistic data from animal models. We don't have much mechanistic data from human studies, and I think that's an interesting place to put some effort now. And that's it.
Thank you very much, Charles. That really gets us off on a good footing. I think we'll go through the rest of our vicious cycle and bring that to some coherence, and then open up for questions and answers. So with that, I'd like to ask Dr. Claudio Lanata, who's also an alumnus of this institution. Claudio got his MPH here in 1983. and has done great work since that time. And of course, comes from the Institute of Nutrition in Lima. So you have two institutional homes here.
Thank you. Thank you, Keith. Really an honor and pleasure to be here. Yes, I was a student in this room. Several of you were my teacher. And it's really great and an honor to be here. So let's start this presentation, and I'm going to end the cycle and talking about infection as a risk for reduced nutritional effect. So as soon as the slides are coming. Yeah, it's coming.
Oh, it's coming.
Yes. All right. Thank you. Okay. This is Peru, and just to bring you a bit, a nice celebrity party in the highlands of Puno. And this is the place where George's mother came. So George has a combination of US and Peru, and it's a nice summary that Keith gave us about him. And they talk about George in nutrition, but also about his very, accents in his opinions. So he got into politics a lot, and as you can see. And thanks to his political connection, he was able to give us this beautiful institute, because he convinced the Peruvian government to donate to us the piece of land when we built this Nutrition Research Institute, where we are working. And this is George. Last visit to our place, that's George, and that's Mary Penny, our current director, Hillary Creed, that's Ron Kleiman, who is member of the advisory board, with Ken Brown, who also was part of Hopkins before, and the whole crowd of the Instituto in honoring George in one of his last visits. And I also, before going into the topic, want to thank George, because I didn't have any idea about the Instituto when I was studying medicine in Peru, as many times it happens. So I had to take I was doing epidemiology, and there was a course about reading in nutrition, and I thought, my gosh, I always hear nutrition, and it's good to have some idea before going back to Peru. So let me take this course, elective course, reading in nutrition. But you have to have the okay from the professor in charge of the course, and you have to get an appointment for him to give the okay for you to sign in. So I walk into George's office, like this picture, and he talked to me in perfect Spanish and Peruvian Spanish, and knew all my family. You are from this, my God, I'm completely surprised. And that's how I got to know the Instituto, where finally I'm working. So thanks for that course. It changed my life. All right. Getting back to this fantastic work that was done in that really breakthrough review 40 years ago. That review has been concentrated looking on effects on nutrition from diseases well known at that time, typhoid, TB, measles, and a whole series. I mean, the really very exhaustive review that you did in every little inch that you can measure in terms of different animals and virus, protozoal, helminthics, bacteria, everything. But as Charles has said, mostly were based on clinical studies as well as laboratory animals, very detailed studies looking on metabolic, nutrient balance, and some micronutrients. But there were no longitudinal studies. I mean, EPI was not around much on those times. So very few really long-term studies carefully done were conducted at the time of that review. But I pick a particular topic, and I'm going to build on that. One of the subjects they review, which was presenting in the monograph as a negative finding, was that they were describing the growth of children where they were given antibiotics. But they were frustrated because when they stopped these antibiotics, the child go back to not growing well. But when the child were given those daily antibiotics, they show that they improved their growth as compared to placebo. Interestingly, when they changed to other type of antibiotics like penicillin, that effect didn't exist. So they were already touching the composition of the microflora in the gut and what are the consequences of having only junk in our gut, as I will discuss later. And I think they were really touching this bacterial overgrowth that exists in the gut of children in developing country, which is now called by many the tropical barrier for delivering good antigens to the gut in the area of vaccinology. We are dealing with that. And indeed, children in poor countries are permanently exposed to unsanitary conditions. They eat, drink, and swallow millions of microorganisms daily in their daily diet. And that creates a burden on nutrition because the body has to fight with all of these visitors and keep them under control. And we will see how it happens. And let me start by a wonderful work that William Sheckley did in a cohort in Peru with a group of, some of them are here, showing how the first encounter of a human body with a parasite like Cryptosporidium affected growth on children, and they, once they were infected, and if they also in those infections had diarrhea, they gained less weight as compared to controls. But when children were infected without having diarrhea, they also had an effect, was less marked than when diarrhea was around, but given the fact that the asymptomatic infections were much more common and frequent, the total burden on nutrition was more important, the asymptomatic component, than the diarrhea-associated burden with crypto. They also show that the effect is long-lasting, particularly if the child is infected early on in infancy and if they were already stunted at the time of their first infection. So this has been validated. There has been study in another cohort in Brazil showing more or less the similar result. But very interesting, the effect was different by species. So the gut is not always treating everybody the same. So in the 0 to 3 months after infection, both of these types of Cryptosporidium had the same effect. But in the follow-up period of 3 to 6 months after infection, only one subtype of crypto had a long-lasting effect on nutrition. And again, in this cohort in Brazil, the greater effect on nutrition was about asymptomatic infections than symptomatic diseases. In another study in Guinea-Bissau, very interestingly, very similar weight loss associated with crypto infection, but again, a gender issue. So our gut also reacts different by different parasites, but gender seems to be also an important variable to take care of. And I don't think in the monograph gender came that strong as a potential covariate to be looking into the effects. And this is the crypto. And there's still a lot to know how exactly it works, but one of the theories is that it covers up your gut surface in a way that it will not only create some degree of inflammation, but it just by mechanical effects, it may create malabsorption and fighting also with the nutrients, having this effect in the gut. And I'm going now to pick in a very close relative, is the Giardia. And I'm willing to bet if I make a little test on all of you, I will get majority of you saying that Giardia is the same. It's a pain in the neck back that creates a lot of problems. Again, in this cohort done in Peru, obviously from birth, many children are infected with Giardia, reinfected. Some of them could be infected for a year, excreting weekly the parasite, but it doesn't affect growth. And it did not have any risk of diarrhea, not at all. We have another study we did with Bob Gilman, who is sitting here, and it's unpublished. It's one of my tragics in life. A lot of papers that need to be written, and it's nice to be in the school because we welcome students who have abilities to do data analysis because we have a lot of data that needs to be published, so you're welcome. We have the same effect. But interesting in that study, in contrast to the one that is being published, is that we also look on fat malabsorption, glucose malabsorption, and several other indicators of Giardia, as it was thought, the mechanism by which Giardia could be producing nutrition and impact. And none of those indicators was associated with Giardia infection. So how could that happen? Because in the same city, if I take a group of gringos who hasn't left the US and bring them to Lima, one of their nasty diarrhea they get by traveling there is caused by Giardia. So it's not that the Giardia that is there is not pathogenic. It's something happening that makes a difference between these two. So this is hypothesis. It seems to be that early exposures, early infections in children may activate the immune system in order to make them tolerant to this pathogen. While if you acquire this infection later in life, then you are really susceptible and then you develop Blount disease. And there is animal data endorsing this. There is a rat model in which if you can infect the rat very early in life, nothing happens to them. They keep no disease, they grow normally. But if you take the same rat and infect them after a critical age, you may even not only make them sick, but kill them by the same strain of Giardia. So very interesting change in age related to this mechanism of tolerance. And that brings me to how, Charles was talking about mechanisms, and we are, I think, moving forward on that concept. And there's a whole now new wind of attention to the mechanism of gut immunology. And in this area, there's a new key player, number 10 in soccer, which is dendritic cells. And it looks like these cells are the key player in order to keep some kind of order in the gut. They are involved in capturing everything, food. Every foreign antigen that is delivered is captured by these dendritic cells, and they have like a computer say, oh, I know them, fine, not to worry, don't overreact, and keep them under control. So that mechanism of capturing that works even in the crypt or in the patch, in the Peyer's patch cells on M cell dependent uptake of different antigens, the dendritic cell seems to be very important, and they do condition imprinting and this tolerant effect in immunology. So they are very early on activated, and there has been studies in neonates showing in developing countries showing how quickly they start acting in the gut on developing countries, particularly when they are exposed to so many pathogens that are being acquired by these individuals. And they recognize them, they kind of, okay, you work in this neighborhood, I know you now, I keep you under control, don't cross the line, I will keep you under control, but I won't overreact like a disease. So very interesting role, and I think we need more to learn about them. And obviously, vitamin A is crucial in that role. So dendritic cells do have a key relationship with vitamin A, and I think Charles alluded to that for their correct function. They also travel all over the body, bringing the antigens they have captured and delivering to the appropriate effective cells, either in the spleen or in the mesenteric nose, in order to build this appropriate immune response. So this is fine for immunology, but what about nutrition? This has a cost, obviously. All this work, it has been exhausting. And so I think the body is really actively involved in keeping all these foreign antigens, food, microorganisms under control. And immunotolerance is not really an immune suppression that we used to think a few years ago. It's just that they are so activated, so early activated, that the response creates that tolerance. It just maintains them under control. And it's fascinating. I mean, in children in developing countries, it has been measured that they are producing 3 to 5 milligrams of secretory IgA per day in the gut in order to fight all these infections. And there is different responses in the upper gut and in the colon. And if you look on the types of dendritic cells sitting in the small bowel or in the large bowel, they are quite different species. So, because they have to deal with different bacteria and different environment. That also explains why the gut has such a strong immunological memory, able to respond to antigens, even though when you are looking on serology markers, they have disappeared a long time ago. They do respond, and I think in the cholera vaccine trial in Bangladesh has been a very nice example of that. And what I'm trying to make in this is that it is an important nutrition burden that needs to be studied and quantified. I think this is something that we need to keep making progress to understand the cost for nutrition of living in poverty. And I took Abdullah's review on this graph just to show that obviously poverty is very complicated, relates to undernutrition by several mechanisms in which infectious disease, low birth weight, and inadequate dietary intake, by that not only on the nutrition quality, but also in the microbiological content. And we are working in Peru now on food hygiene and how in order to bring a better diet to children, to bring down the bacterial exposure in their daily intake, because it has a problem. And I think everybody has heard about Leonardo Mata's famous studies in Santa Maria Cauqué, where he was one of the first in the group there to demonstrate that this line was the growth curve on a normal child in the reference population. This was the group of children they studied, and this dotted line was an individual child that obviously ended up dying, and they were very nicely showing how at the beginning they were kind of protected, mostly because the amount of breastfeeding these children were receiving was massive at that time. But as soon as they start getting weaning foods, they start getting a lot of diarrhea and respiratories and a whole bunch of diseases that end up, in some of them, killing them, and in many of them have really a very low growth curve, keeping them undernourished and also psychomotorly retarded. So diarrhea was recognized very early then, and it has been always quoted as one of the best examples of an infection causing undernutrition. Abdullah and Baki and Bob Black review 23 prospective studies looking in this issue. 21 of the 23 show an impact on weight. The 2 that did not were done in Taiwan and Australia, so they probably were having a different diet quality than the rest of the developing country studies. From 30 studies that had data, you can see the weight loss attributable to diarrhea, around 40 grams of lost weight per day. 13 studies looks on length. 4 didn't show any effect, but 8 had again around 0.08 millimeters of length lost per day. But Abdullah couldn't do a meta-analysis. Why? Because these studies are very hard to compare. There are different case definitions, different methods, different mechanisms by which the different variables were taken, and they were analyzed differently. So very hard to combine together. And that prompted, again, William Sheckley, who is sitting here, to I don't know how he did it, but he was able to really grasp 9 longitudinal data, original databases from different studies, from 5 countries that had frequent anthropometry and diarrhea surveillance. And they analyzed incidence and prevalence and look on the amount of disease that the children had in the first 2 years of life to predict stunting at 4 months of age, controlling for several variables. And you can see here, it is a linear relationship. The higher number of episodes of diarrhea, the higher your chance of being stunted at 4 months of age. And the same thing, the more prevalence, number of days of your life that you have diarrhea with, also the higher the chance of developing stunting at 4 months of age. And this is the odds of becoming stunted at 24 months of age if you have an increase of 5 episodes of diarrhea more per year in the first 2 years of life. It's not huge, it's 1.13, but significant. Not too different if you look on prevalence. Interestingly, that study also showed beautifully that this catch-up growth that has been described doesn't exist that often in terms of length. And very few children who were stunted at 6 months of age recover at 24 months of age in these 9 studies. And this was done in analysis that controlled for the regression towards the mean, which was a major problem in the original studies that how to interpret this data. And this brings me back to a diagram that our friends put together in the 1968 monograph, in which they were considering this is total body nitrogen, and this is the nitrogen balance. So they were thinking, okay, you have an infection here, you will start losing weight, and your total balance of nitrogen is gone, and this is because you're losing by consuming a lot of your energy. And then if you catch up, you recover from the infection and you're having a good diet, you will be able to build a positive balance of nitrogen, and therefore your total body nitrogen will recover. These are normals. If you are really malnourished, the C letter, you will lose body mass, and you may barely catch it. I think they need a forward graph here, because some of them don't go back to the baseline. As Leonardo Mata diagram show, they continue coming down. But they also thought that some people could lose, but then gain and keep gaining a positive nitrogen balance, so they will be able to really catch up and recover their malnutrition. And I think that is what is being now looked. This doesn't seem to be that often happening in developing countries. Why? Because of quality of diet. And this is a nice study that Bob and Guillermo Lopez de Romagne and Ken Brown did in Lima, in which they were studying children who were having different types of quality of diet normally, not during illness all the time. So, and they look, all these children who were having a better amount of calories per day, per kilogram, if they have diarrhea, no matter what was the prevalence of diarrhea, there was no impact on growth velocity. But if they were on a middle or lousy diet, the more diarrhea, the less weight gain they were having. So you can, in a way, by this catch-up phenomena that was very, very nicely published in the concepts in that monogram, you really can revert this metabolic effect on diarrhea and nutrition. Mechanisms has been studied, reduction of energy consumption because of poor appetite and vomiting. There was a major grant from USAID which was called Feeding During Diarrhea, because in the 80s, some people thought that the mothers in developing countries were so stupid that they were going to dilute the diet when the child has diarrhea and really support him with a very awful diet. Thank God didn't happen. Well, normally the mother is recognized that the child has lost appetite and they reduce the volume of food, but they don't change. They don't change and give a particular recipe that is very lousy for nutrients. So that doesn't seem to be the case. There is reduced absorption of nutrients because of destruction of the cells, increased motility and malabsorption that we were discussing before. Some issues on permeability to test sugar that brings potential chronic disease, because some of this diarrhea, by that increased permeability, expose antigens to the inside the lumen, into the interstitial that shouldn't be there. And suddenly triggers a whole of reactions and chronic inflammation that was unaware before. There is a lot of loss of nutrients, particularly zinc, copper. Vitamin A is a whole chapter of vitamin A in the urine and all the metabolism of vitamin A and infection, as well as increased utilization. I think in diarrhea, enterotoxigenic E. coli, rotavirus, and dysenteric illness are best examples, as well as persistent diarrhea as good how these diseases are more serious. Changing gears, what about respiratory infections? Are they same? And here we have much less data. There has been much less number of studies on respiratory disease looking into this interaction. Again, in the review that Abdul and Bob did, they only found 8 studies, and 4 of them has found an impact. The others did not. And not of any respiratory infection, but mostly the lower, more severe. Some of them with similar amount, a bit greater loss of weight per day than diarrhea studies, but only 2 studies. In terms of linear growth, 3 or 5 studies, again with more severe disease, had an effect. They have documented that there's reduction of food intake mostly because of fever and severity of illness. Obviously, because respiratory illness not necessarily destroys the gut tissue. But we need more studies. And I think that's the major message on respiratory diseases. I don't think this interaction has been looked that carefully with this type of disease. Obviously, TB is an exception. It was included in the review, long recognized as a cause of severe wasting and negative nitrogen balance. Interestingly, chemotherapy improved nutrition status. especially when good nutrition support is given. But there's no more studies showing that also in that infection, in chronic infection like TB, there is an anabolic block that develops in some patients that makes them refractory even to nutritional support. So again, there is a need to understand better this mechanism by which some of these chronic infections do have a much more strong effect on nutrition, and they become refractory for a time for nutrition rehabilitation. And obviously, it was not existing in the 1968, but the HIV/AIDS infection and TB has made things much more worse for nutrition. Measles was recognized importantly as a cause of kwashiorkor in undernourished children in the review. Anorexia is refusal to eat, catabolic effect, protein-losing enteropathy was described. In the latest years after the review, I think they recognized that acute and persistent diarrhea do occur long time after the initial episodes of measles, because there is a persisting immunosuppression in these patients, which was not known in the 60s, and a whole of interaction with vitamin A that makes the vitamin A a key element in the treatment of measles cases. Malaria was also mentioned. Very few studies in the 68 review. But I think there's nice data now that has been emerging from trials, particularly preventive trials like pyrimethamine and chloroquine prophylaxis versus placebo that has shown that children in Ghana, Gambia, and Nigeria increased growth when they were given this prophylactic medication, not only because of the medication, because the insecticide-treated bed nets has the same effect. So just avoiding this infection with the mosquito do have a nutrition benefit. And a very nice study in Vietnam, not just a trial, but an effectiveness type trial, because it was done under programmatic conditions over 5 years. They also has seen a very positive nutrition effect by controlling malaria. And this is my last. Obviously, HIV/AIDS makes everything look awful. The effect starts intrauterus. It's a high risk of intrauterus growth retardation and low birth weight. Children in Africa that are infected at birth, even if they're asymptomatic, they do have a very bad growth compared to the non-infected children. There are several mechanisms directed by the virus, as well as the HIV-induced secondary infections. And this wasting seems to be related again to all these inflammatory and immunological mechanisms that Charles was describing, especially these interleukins that seems to be very crucial for anorexia, and several macronutrient deficiencies also are being described with HIV/AIDS in these children. So in conclusion, I think since 1968, we have now a lot of well-conducted epidemiological studies and clinical trials that has documented very clearly the importance of infections on nutrition in developing countries. But I think with few exceptions, I could say that these effects may be transient and not lasting if these children are well fed. And I think that's a very important message, because I think we have a lot to do by improving dietary intake of children in developing countries and help them deal with this burden of infectious and non-infectious agents that have to be fighting all the time. So the nutrition costs of living in poor, unhealthy environment with high intake of microorganisms and asymptomatic infections, I think we need to look at more carefully and measure how much cost the body is paying to deal with these conditions in poor environments. So I want to finish by thanking Navin, Carl, and John for stimulating 40 years of research. Very exciting, and I'm sure that this will continue, hopefully in the next 40 years, by many of you guys sitting here doing key research to finalize understanding all these interactions. Thank you very much.